sticking it out. We'll get you to the poster session here soon. I guess some of you saw what I was going to say for the slide already anyway, but my name is Backenstose, and I'm a PhD candidate in the Coregonus lab at the University of Buffalo. And today I'll be presenting an element of my dissertation work that has a focus on two lineages in the Great Lakes, the Ciscoes in the genus Coregonus and the lake Charr. And both of these lineages have undergone radiations and constituting some of the largest freshwater bodies in the world, the Laurentian Great Lakes, provide a lot of habitat. And not only do they have a lot of surface area, but they are also very deep. In the right panel, you can see a contour map of Lake Superior, as if you were looking from west to east. And then you have the depth profile. And I don't know if you can see at the bottom, but it says 400 meters, and at its deepest point, it's around 1,333 feet. And this depth gradient provides a lot of ecological opportunity for divergence. And with great depth comes great ecological opportunity. And I promise I did not steal that from the plenary this morning. I had it pre-planned. With this ecological opportunity, we have both of these species, both of these lineages separated into species such as the Ciscoes on the left here, the Artidae, which hangs out between 5 and 90 meters, the Hoyai between 55 and 90 meters, and then the Kaiai greater than 95. And you can also see some of the phenotypic variation within this group that comes with depth, notably the large eye in the Hoyai and Kaiai there. The other lineage, the lake Charr, I've displayed two ecomorphotypes here, the lean and the Ciscoette. The lean is the shallower, prefers shallower depths, and the Ciscoette prefers deeper depths, and Chris just explained some of the different phenotypic variation that comes with the Ciscoette morph. I would like to thank NYCAFS for setting up this event and providing a theme that fits really well into my area of research. And with the theme of then and now, I'm going to be focusing heavily on the then, with a look into the historical demography of these two lineages. And with this look into the past, I'm going to see how it relates to their coevolution. And I've displayed here a major environmental event that influenced the evolution of both of these lineages. And that is the last glacial maximum, which occurred quite a few thousand years ago. And with that in mind, and with this talk, I will be addressing the questions that are posed here. When did these species and ecomorphotypes diverge? Does prey diversification affect predator diversification? And what are the underlying genetic and genomic variation, and is that associated to phenotypic variation? So adding some more context to our timeline here, I did mention the last glacial maximum that occurred 19 to 33,000 years ago, where the Laurentide ice sheet covered what we now know as the Great Lakes. And also a population collapse of Ciscoes that occurred in the mid-1900s, caused by overfishing, habitat degradation, and invasive species. And lake Charr followed shortly thereafter. Both these events influenced the evolution of these species and their population sizes. And I'm going to display here how we can reconstruct these through time. And so the previous talk highlighted some of the benefits of whole genome sequencing. And I'm going to elaborate on that here, and specifically in how we can reconstruct their demographic histories through time. And somewhat unbelievably, we can actually utilize the whole genome sequencing from a single individual to do this. And we do that with coalescent theory. So within each genome, there are large numbers of loci that are separated by via recombination. And we can track the evolutionary histories of these loci. On the left, we have a single genome here with the loci separated by the double gray lines. And you can see there are differences in the alleles at each locus represented by the blue and red dots. By tracking coalescent events of these alleles, we can see how many occurred within a given time period. Sorry. And if there are more allelic differences at a locus, we can conclude that that happened further back in time. And if there are fewer, we can conclude that those are more recent in time. The software we utilize called PSMC, can take this information and reconstruct effective population sizes through time. And these estimated changes in effective population size can help us understand our first question of when these species actually diverged. So I'm going to jump right in here, and we used whole genome sequencing of 4 Cisco species that I have presented here, 3 from Lake Superior, the Artidae, the Hoyai, and the Kaiai, and 1 from Lake Nipigon, which is north of Lake Superior, Coregonus subricanus. We sequenced them to about 18x coverage, and we mapped them to a draft reference genome we have for Coregonus Artidae. And then with PSMC, we estimated their effective population sizes through time. And I'm going to walk you through this figure here. On the x-axis, we have time on a log scale. In the top panel, we have global mean temperature. And in the middle panel, we have those estimated effective population size trajectories. And each line represents a single sample of a given species. And then in the bottom panel there, we have images related to the extent of glaciation that occurred after the last glacial maximum as the glaciers were receding. So what you can see here is that during the last glacial maximum, the effective population sizes for Ciscoes were at their lowest point. And that makes a lot of sense to us because that's when they were pushed into glacial refugia. And then following deglaciation, they expanded rapidly in population size. And this rapid expansion kind of makes it difficult to see what is going on with these individual lines. So I've zoomed in here, and if you follow the individual lines, they actually coalesce at this certain point, sometime around 60 to 90,000 years ago, favoring the latter. And this is likely when a common ancestor diverged. If the lines were in tight concordance until, say, the last glacial maximum and then broke apart, then we would think that they diversified after the last glacial maximum. So we can conclude here that, and we estimate that the diversification of these species occurred prior to the last glacial event. And following this divergence, and with the occupation of the Great Lakes, these species evolved into a complex of, with phenotypic and functional diversity that was seen when these fishes were classified. And so now that we have this time range, we kind of naturally ask, what was the environment like when this diversification occurred? Diving into the geologic literature, we found that this 60 to 90,000 year mark was during what's called marine isotope stage 5. And that's just a fancy term for a very warm interglacial period. So actually, the Great Lakes region was not covered with any glaciers at that time. The precursors to the Great Lakes were present, and they were likely of significant depth for some diversification to occur, like we see here. So now that we have estimated effective population size for Ciscoes, now we're about a native primary predator. The addition of lake char in these analyses gave us the opportunity to look at what the diversification of prey species can do to a predator species, and what are the demographic shifts associated with that. There's often a lot of emphasis on evolution of predator-prey complexes in terms of evolutionary arms races, but not many people look into what happens when a prey species diversifies and what happens to the predator. So since there is a genome for this species, from Seth Smith et al., as Christopher mentioned, and multiple ecomorphotypes of this predator species as well, we sequenced 2 different ecomorphotypes, the main and the Ciscoet, and then ran PSMC and plotted them here with our Ciscoes. And overall, you can see that the effective population sizes are lower, which makes sense for a predator species. and that the low point also occurred during the last glacial maximum. And then, most importantly, there's a lag time in expansion that followed the Ciscoes. So the Ciscoes may have invaded new niches, and Lake Charr may have diversified to follow them. There. So in this diversification scenario, we have species that overlap with depth, such as the lean and artidae, and the Ciscoet and hoyai and kaiai. And we have some contemporary diet studies that reflect this overlap, with leans feeding on artidae and Ciscoets preferentially feeding on hoyai and kaiai. So the final set of analyses I would like to leave you with pertains to fixation of genes within the Cisco species complex, and specifically in Lake Superior. We took the Lake Superior whole genome sequencing data and conducted FST analysis, which is a measure of genomic variation. A 0 value indicates complete panmixia or sharing of genetic material, and a value of 1 indicates no sharing. So I've displayed the values here in a Manhattan plot. This is a global FST for all the superior species. And you can see here there are a few areas of high FST SNPs, but I want to point out this one specifically on chromosome 3. So this could be a structural variation, as Christopher mentioned in the last talk, such as an inversion that could have some co-adapted genes within it. And when we zoom in here, we can see that this is a 12 meg base region that is depauperate of low FST SNPs. The dark lines indicate a 99% threshold, and then we have a moving average along the bottom. And this is interesting because, as Christopher mentioned, these inversions can disrupt recombination and protect co-adapted alleles protect co-adapted alleles that can be linked to specific phenotypes. We did some investigation into what's in this region, and we found some interesting genes related to eye development and circadian rhythm and light stimulus genes. So when we saw this, we also made comparisons between pairwise comparisons between members of the complex. And I have here an artidae and kaiai FST plot, Manhattan plot. And I want to point out this area here on chromosome 33. And we think this is a kaiai specific area that has another high FST peak, which could be an inversion. And there are also eye genes and other germ cell development genes on there as well. So that's really interesting. And just to conclude here, some of the questions: when did these species and ecomorphic types evolve? Specifically for the Ciscoes, we estimate somewhere between 60 and 90,000 years ago. Does prey diversification affect predator diversification? So in this scenario that we have laid out here, we have what's called an upward adaptive radiation cascade, where a radiation in the prey species facilitates a radiation in a predator species. And what's the underlying genetic and genomic variation that's associated with phenotypic variation? We have these putative inversions with possible co-adaptive gene complexes, and we'd like to explore this more with some of the methods that were highlighted in the previous talk. And with that, I will acknowledge everyone that was involved with this project, past and current members of the Crab Lab, Dr. Louis Bernaches and Eric Nomadu for help with scaffolding the genome, collaborators at USGS, Daniel did a lot of work on this project, and also Anders Nyland, Eric Bergerland, Dave Montgomery from OMNRF, and funding agencies. And with that, I will take questions, and if you don't get to me, my contact information is in the